Usually when we talk about a medical diagnosis, there's this underlying expectation of precision, right?
Absolutely. It feels almost like engineering most of the time.
Yeah, exactly. Like you break your arm, the X-ray shows that jagged height line right through the radius, and the doctor just points at the screen and says, well, there it is.
Right. It's entirely binary. I mean, it's either broken or it's not broken. And it's a clean, visible categorization that we as clinicians find incredibly comforting.
It is comforting,
Yeah.
but then, you step away from the acute trauma bay and you get into the world of systemic bone metabolism, and suddenly that X-ray machine is, well, it's effectively useless.
Yeah, you're flying blind, essentially.
Exactly. We're looking at a diagnostic landscape that is, honestly, it's murky. And nowhere are those waters muddier than in the basic sciences you have to master for the FRCS exam.
Oh, the FRCS basic science. It's a nightmare for so many candidates.
It really is. And the problem with basic science for the trauma and orthopedic sports isn't that it's, conceptually impossible to grasp.
Right. It's not quantum physics.
No, it's not. It's that it arrives as this sheer avalanche of tiny, seemingly disconnected facts. Like one cell does this, one hormone raises calcium, another one lowers it.
And the exam just sits there quietly waiting for you to mix up two molecules that have, like, almost identical names.
Oh, they love doing that. And the brutal truth is that you cannot simply out-memorize this material.
You really can't. I mean, trying to brute force 200 loose facts into your head, it just won't work, especially when you're trying to revise after a 24-hour on-call shift, right?
Exactly. What actually stays in your memory is a story. What survives the stress of the exam room is a solid logical structure that you can hang those individual facts onto.
Which is exactly why we're doing this.
Right. That is exactly the mission for this deep dive. We are targeting FRCS basic science, specifically the notoriously dense module of bone cell biology and calcium homeostasis.
It's a heavy one, but we've got you covered.
We do, and we know exactly where you are right now. You're probably driving home from the hospital, or, you're doing the washing up, or you're staring at the ceiling, trying to squeeze in some high-yield revision before your head hits the pillar.
So here is the promise for the next hour. You do not need notes.
Put the pens down.
Seriously, put them down. Keep your eyes on the road. Let the concepts do the work for you.
Yeah, we will not reference a single diagram. We won't point to any figures. Everything will be explained verbally.
And every single critical number, every list, and every tricky exam discriminator will be repeated twice.
Twice, because we know you're distracted. By the end of this, you will be able to trace one logical thread from what a bone is actually made of, all the way to which hormone raises your phosphate levels and which one dumps them into your urine.
To give you that structure, though, we really need to establish the overarching spine that runs through this entire biological system.
Okay, lay it on us.
Yeah.
What's the core rule here?
Well, if you internalize this one concept, the pharmacology and the pathology just fall into place. Bone is a permanent, relentless tug of war.
A tug of war.
Exactly. It's a never-ending fight between building up and breaking down. Every single hormone, every drug, and every pathway we discussed today is doing nothing more than picking a side in that tug of war.
Ah, okay, so it's just picking a side and shouting instructions at one specific cell to make it happen.
That's it. That is the entire secret to this module.
A permanent tug of war.
Yeah.
Let's hold on to that image. But before we can look at the two teams pulling the rope, we probably need to understand the rope itself, right?
We definitely do. We need to look at the materials.
Right. So if I were to take a femur, cut a perfectly clean slice through the cortex, and put the raw ingredients on a biochemical scale, what are the exact proportions we are looking at? Because I know the exam loves to test the precise recipe of bone.
Oh, they test it relentlessly. So the recipe has 3 main ingredients, and these percentages are highly examinable.
Okay, I'm listening.
Roughly 70% of that cortical bone is mineral.
70%. Wow, okay.
Yep. And that mineral is specifically hydroxyapatite, which is it's a complex crystalline lattice of calcium and phosphate.
Hydroxyapatite, got it.
Right. And that 70% mineral component is what gives bone its incredible stiffness. It's what allows the skeleton to resist being crushed under the compressive weight of gravity.
Okay, so 70% is the hard stuff. What's the rest?
Then you have about 22 to 25%, which is the organic matrix.
22 to 25% organic matrix.
Exactly. And the final sliver, making up just 5 to 8%, is simply water.
Okay, let's lock those proportions in for anyone who just, merged onto the motorway. 70% mineral, 22 to 25% organic matrix. 5 to 8% water.
Perfect.
I'm going to say that one more time just to cement it. 70% mineral, 22 to 25% organic, 5 to 8% water. Now, organic matrix sounds like a generic biological soup.
It does sound like that, but it's really not.
Right, it's actually a highly specific engineered structure, isn't it?
It is overwhelmingly dominated by one specific protein. Of that 22 to 25% organic matrix, roughly 90% of it is type I collagen.
Wait, 90% of the organic part is just type I collagen.
Yes, this is a number you absolutely must tattoo on your brain. 90% of the organic matrix is type I collagen.
So if 90% is type I collagen, what is its mechanical purpose? I mean, if the hydroxyapatite mineral is handling the hard compressive forces, what is the collagen doing?
Well, where the mineral resists crushing, the type I collagen does the exact opposite. It provides tensile strength.
Tensile strength, so it stops it from pulling apart.
Exactly. It gives the bone its springiness, its ability to bend slightly under a load, and its resistance to being snapped.
That sounds exactly like reinforced concrete in civil engineering.
It's the perfect analogy, honestly.
Right, because if you pour a massive concrete pillar, it can hold up a skyscraper because of its compressive strength. But the second an earthquake hits and the building sways, plain concrete will just snap because it has zero tensile strength.
Precisely. Which is why engineers bury steel rebar inside the concrete.
Right, the concrete takes the compressive squeeze and the steel takes the tensile pull. So in our skeleton, the hydroxyapatite mineral is the concrete and the type I collagen is the steel rebar.
That analogy holds up perfectly under biomechanical scrutiny. The mineral is the concrete, the type I collagen is the steel rod.
It makes it so much easier to visualize.
It does. And this brings us to a fact that examiners absolutely love to use as a discriminator. Nature specifically chose type I collagen for this job because type I collagen is the only collagen that can mineralize.
Wait, it is the only one?
The only one.
So if a multiple choice question tries to bait me with, like, type 2 or type 3 collagen?
You immediately rule them out. I mean, think about the cartilage in your knee joint.
Okay, cartilage.
Cartilage is packed with type 2 collagen, and type 2 does not mineralize under normal physiological conditions. It stays rubbery and compliant.
Because if it mineralized, your knee wouldn't bend.
Exactly. Bone needs to turn into that hard concrete, so it relies on type 1. The unique structural cross-linking of type I collagen is what allows the hydroxyapatite crystals to embed themselves within the fibers. Type I is the only one that mineralizes.
Okay, so if type I collagen makes up 90% of the organic matrix, what is floating around in the remaining 10%? Is there anything in that leftover soup that an FRCS candidate actually needs to care about?
You don't need to know the entire biochemical soup, no. But you do need to recognize a cast of non-collagenous proteins, things like osteocalcin, osteonectin, and bone protein.
Okay, osteocalcin and bone protein.
Right. We will explore a couple of them in depth shortly because they act as highly specific chemical signatures. They're the markers that tell a pathologist or a lab exactly which cell is active.
But for the basic structural recipe, we just group them into that final 10%. So if I'm a tired candidate listening on a commute, the vital takeaway I am committing to memory right now is the recipe. Bone is 70% mineral, and type I collagen, which is the only collagen that mineralizes, makes up 90% of the organic part.
That is the core takeaway, yes.
I am repeating that to be safe. Bone is 70% mineral, and type I collagen, the only one that mineralizes, makes up 90% of the organic part.
So that covers the materials. Now we have to look at the workforce that actually handles those materials. We need to meet the cast of characters engaged in this metabolic tug-of-war.
Right, if we know what the concrete and steel are made of, who is pouring the foundation, and who is driving the wrecking ball.
There are 3 distinct cells you need to master here. And if you can keep the biographies, the origins, and the daily jobs of these 3 cells straight, a massive portion of this module will unlock for you.
Okay, let's meet them. Who is character 1?
Character 1 is the osteoblast. That is your builder. Character 2 is the osteoclast. That is your demolition crew.
Osteoblast builds, osteoclast demolishes.
Got it. And the third, in character 3, is the osteocyte, which is, biologically speaking, an osteoblast that got buried alive in its own concrete.
Buried alive. Okay, we will definitely come back to that one. Let's start with the builder, the osteoblast. What is the day-to-day function of this cell on the bone surface?
So the osteoblast is an incredibly active protein factory. It manufactures and secretes the type I collagen we just discussed.
Right, making the steel rebar.
Exactly. It also produces those non-collagenous proteins, specifically osteocalcin and bone siloprotein. And osteocalcin is a fantastic exam topic because it is a vitamin K-dependent protein, and it's utilized clinically in blood tests.
Oh, I've actually seen that on the wards.
Mm.
If an endocrinologist wants to know if a patient's bone formation rate is increasing, they look for serum osteocalcin.
Right, exactly, because only active osteoblasts spit it out while they're working.
Makes sense. But producing the collagen steel rebar is just one half of the job. What about the mineral? Does the osteoblast pour the concrete too?
Yes, and this is a crucial single best answer discriminator for the exam. The deposition and mineralization of the bone matrix is handled solely by the osteoblast.
Holy.
I use the word solely because the exam stems will try to trick you into assigning mineralization to other cells, systemic hormones. The mineralization of bone is done solely by the osteoblast.
Solely by the osteoblast. Okay, to dig into the how of that, though, I mean, it can't just be leaking raw calcium and phosphate into the surrounding tissue, or the whole area would just petrify randomly. There has to be a mechanism.
There is, and the mechanism is highly localized and elegant. The osteoblast buds off these microscopic packets from its membrane, which are called matrix vesicles.
Matrix vesicles.
Right. You can think of them as highly targeted biochemical depth charges. These vesicles are packed with an enzyme called alkaline phosphatase.
Ah, alkaline phosphatase. We test for that all the time.
Exactly. When these matrix vesicles are released into the surrounding osteoid, which is the unmineralized bone, and they pop open, the alkaline phosphatase dramatically increases the local concentration of inorganic phosphate.
And that causes the crystallization.
Yes. That specific biochemical shift kicks off the crystallization of the calcium and phosphate into hydroxyapatite.
So alkaline phosphatase is basically the trigger for the concrete setting. And because it's packed in those vesicles, the setting only happens exactly where the osteoblast wants it to.
Precisely. Which is why serum bone-specific alkaline phosphatase is another excellent clinical marker of osteoblast activity.
Right, but to return to the exam rule, if a question asks, which cell mineralizes bone, the answer is always, only solely the osteoblast.
Oh, got it. Solely the osteoblast.
Now, in a dynamic skeleton, osteoblasts aren't just sitting in one place forever, right? I mean, if a runner develops a stress microfracture in their tibia, the body needs to send builders to that exact location. How do the osteoblasts know where to go?
They migrate using a chemical tracking system. That directional movement along a chemical gradient is called chemotaxis.
Chemotaxis.
Yeah. The osteoblasts literally sense a chemical signal in the environment and crawl toward the highest concentration of it.
So they're following a scent trail. What is the scent?
For the FRCS, the classic chemical signal you must associate with osteoblast chemotaxis is IGF-1, or insulin-like growth factor 1. When bone is damaged, IGF-1 is released from the matrix, creating a trail for the builders to follow.
Okay, so the testful fact there is osteoblasts migrate by chemotaxis, and they follow the scent of IGF-1.
That's the one.
That establishes our builder pretty well. Let's look at the demolition crew.
Yeah.
The osteoclast. And this is where a major conceptual trap lies regarding their origins, doesn't it?
It is a massive trap, yes.
Because if I am guessing based purely on logic, I would assume that since both the osteoblast and the osteoclast work exclusively on bone, they must be cellular cousins. Like they should come from the same stem cell parent.
That is the most intuitive assumption to make, which is exactly why the examiners love to test it, because it is entirely incorrect.
They aren't related.
The builder and the demolisher are not related in any way.
Wow, okay.
The osteoblast, our builder, comes from mesenchymal stem cells. That is your standard connective tissue lineage, the same embryonic origin that gives rise to cartilage, fat, and muscle.
So mesenchyme creates the structural builder. What creates the demolisher?
The osteoclast comes from the hematopoietic stem cell line. Specifically, it belongs to the monocyte macrophage family.
Wait, the blood cell line. We are talking about white blood cells.
We are. The osteoclast is fundamentally a highly specialized, hyperaggressive immune cell. It is a giant macrophage.
A giant macrophage that eats bone.
Exactly. It's multinucleated, meaning it contains multiple nuclei because several individual monocytes had to physically fuse together to create this massive cell. Its entire biological purpose is to latch onto bone tissue and resorb both the mineral and the collagen.
That completely changes how you look at the tug of war. The builder team is sourced from the structural connective tissue side, the mesenchyme. But the demolition team is drafted from the immune system, the monocyte macrophage line.
That's right.
They aren't related at all, which must mean they respond to totally different systemic signals.
Their different origins explain exactly why they are controlled differently. We will see how that plays out when we get to the hormones in a minute.
Oh, I can't wait for that. But before we leave the cast of characters, we must address the third cell, the osteocyte,
Right, the osteocyte.
the builder that gets buried alive. Walk me through how that happens.
Well, the osteoblast is an incredibly busy cell, continuously laying down that concrete and steel matrix all around itself. And frequently, an osteoblast will work so fast that it effectively traps itself inside its own freshly poured matrix.
This just builds a wall around itself.
It does. It becomes completely entombed in a tiny cave within the bone, which we call a lacuna. And once it is trapped, the cell undergoes a morphological change. It stops building, it shrinks slightly, and it transforms into an osteocyte.
And once it's trapped in the lacuna, what is its purpose? It can't build anymore.
It becomes the mechanical sensor of the skeleton.
A sensor. How does it sense anything through solid bone?
While it is trapped in the lacuna, it extends dozens of microscopic spider-like arms through tiny tunnels in the bone called canaliculi.
Canalically.
Yes. These arms connect with the arms of other osteocytes, creating a massive three-dimensional neural-like network throughout the entire bone.
So they are literally holding hands through the concrete walls. What are they sensing?
They are sensing fluid shear stress.
Fluid shear stress.
Yeah. When you go to the gym and lift a heavy weight, your bones physically bend and deform on a microscopic level. That deformation forces the fluid inside those tiny canaliculi tunnels to flow rapidly over the osteocyte's arms. The osteocyte feels that fluid shifting, interprets it as mechanical load, and sends biochemical signals to the cells on the surface of the bone, telling them exactly where the bone needs to be thickened to handle the new stress, or, where it can be thinned out because it isn't being used.
That is the cellular mechanism of Wolff's law. The bone remodels along lines of stress because the buried osteocytes are physically feeling the fluid shift when the bone bends.
Exactly. The osteocyte is the mechanical mastermind of the skeleton.
Okay, let's crystallize the cellular origins for the listener. The vital takeaway here is separating the family trees.
Very important.
The osteoblast builds, it is the sole cell that mineralizes, and it comes from mesenchyme. The osteoclast demolishes, and it comes from the monocyte macrophage bloodline. To double down on that, osteoblast builds and solely mineralizes from mesenchyme. Osteoclast demolishes from the monocyte macrophage line.
With the cast of characters established, we really need to look closer at the actual mechanics of demolition.
Let's get under the microscope and look at the underside of the osteoclast. Because if you want to understand how osteoporosis drugs work later, you have to understand the specific machinery this cell uses to destroy bone.
You do. And the exam usually breaks this down into two distinct anatomical features on the cell membrane.
Okay, what are we looking for?
If you view an active osteoclast on a bone surface, the side touching the bone has two highly specialized zones that perform two completely different jobs. One is called the clear zone, which is also known in some texts as the sealing zone, and the other is called the ruffled border.
The clear zone and the ruffled border. Let's break down the mechanics. What is the clear zone doing?
The clear zone is the attachment mechanism. I want you to imagine the massive osteoclast as an industrial suction cup.
Okay, a suction cup.
The clear zone is the thick rubber rim of that suction cup. It clamps down onto the bone surface with immense force, creating a perfectly sealed, watertight, isolated microenvironment underneath the cell.
And how does it physically grab the bone?
To achieve this grip, the clear zone uses specific transmembrane anchor proteins called integrins.
So the clear zone is purely about clamping down and creating a seal. It doesn't do the destroying itself.
Correct. Clear zone equals attachment. It clamps using integrins. Once that watertight seal is established, the second structure goes to work.
The ruffled border.
Right. Inside that isolated pocket, the cell membrane becomes highly folded and frilly, massively increasing its surface area. This is the ruffled border, and the ruffled border is an acid factory.
An acid factory. How exactly is it producing acid? Is it just dumping hydrochloric acid like the stomach?
No, it utilizes an enzyme system called carbonic anhydrase.
Carbonic anhydrase.
Yes. This enzyme takes carbon dioxide and water and rapidly converts them into protons, which are hydrogen ions and bicarbonate. The cell then actively pumps those protons straight across the ruffled border and into the sealed pocket.
And what happens to the pH in there?
The pH inside that isolated space crashes down to roughly 4.5. It becomes incredibly acidic.
And what does that acid do to the bone? I assume it attacks the concrete.
Exactly. The acid dissolves the hydroxyapatite mineral. You have to demineralize the bone before you can degrade the collagen. Otherwise, the enzymes can't reach the protein fibers.
That makes perfect sense.
Once the acid has melted away the mineral, the ruffled border secretes proteolytic enzymes. The most famous one for the exam being cathepsin K, which digests the leftover type I collagen. But the defining feature of the ruffled border is that acid pump.
So to pair the buzzwords, the clear zone clamps using integrins. The ruffled border resorbs using carbonic anhydrase.
That is the crucial pairing.
Let's repeat that pairing for the listeners. Clear zone clamps using integrins. Ruffled border resorbs using carbonic anhydrase.
Now, we must pause here because this specific topic contains a notorious pitfall for candidates revising with certain online resources.
Oh yes, I know exactly what you're talking about. I've seen a wildly circulating question bank in the FRCS quiz circuit that gets this dead wrong.
It does.
We need to explicitly correct this on air. The question stem asks, which part of the osteoclast is responsible for attachment to the bone surface before resorption? And the provided answer key in this popular quiz says, ruffled border.
It is entirely biologically incorrect. And the most frustrating part for a trainee is that if you read the explanation text below the wrong answer in that very same quiz, the text correctly describes the clear zone attaching via integrins.
The answer key contradicts its own explanation.
It does. It is a typo that has survived for years and caused endless confusion.
Let's dismantle why that's biologically impossible. If the ruffled border was the attachment point, it wouldn't be ruffled, and the cell would be pumping acid directly into the open extracellular fluid instead of a sealed pocket.
Exactly. The seal must come first. So if this exact concept comes up in your real exam, trust the physiology. Attachment is the clear zone. Full stop. The ruffled border is for resorption. Ignore the typo in the quiz bank.
That clarification alone is going to save someone a crucial mark. Now, before the osteoclast can even utilize its clear zone to clamp down, there is a fascinating sequence of events that has to occur on the bone surface. Because an osteoclast cannot just wander over and attach to any random piece of bone at once, right?
It cannot. This is a subtle but heavily tested point. Resting bone, meaning bone that is not actively being built or destroyed, is covered by a microscopic, very thin layer of unmineralized osteoid.
Unmineralized osteoid.
Yes. It acts like a protective biological tarp. The osteoclast's integrins are designed to grip mineralized matrix. They cannot grip unmineralized osteoid. So as long as that tarp is there, the osteoclast is locked out.
If the demolisher can't get through the tarp, who removes it?
The builders do.
The builders.
Yeah. Sitting on the surface of resting bone are cells called bone lining cells. These are essentially flat, sleepy, retired osteoblasts.
Okay, retired osteoblasts.
When a systemic signal dictates that a specific area of bone needs to be resorbed, these sleepy bone lining cells wake up. They secrete an enzyme called collagenase, which chemically digests that thin protective tarp of unmineralized osteoid.
So they clear the site.
Furthermore, these lining cells physically contract, pulling themselves back to expose the bare mineralized bone underneath. Only after the lining cells have cleared the surface can the osteoclast move in, clamp its clear zone, and begin pumping acid.
That completely reinforces the tug-of-war narrative. The demolition crew is powerless until the builder lineage actually prepares the site and grants them permission to work. The osteoblast lineage is pulling all the strings.
That concept that the builder controls the demolisher is the fundamental rule of bone biology.
Which gives us our vital takeaway for this anatomical deep dive. The clear zone clamps via integrins, the ruffled border resorbs via carbonic anhydrase, and attachment is always the clear zone, regardless of what a faulty quiz tells you.
Always the clear zone.
Repeating that core pairing again. Clear zone clamps via integrins, ruffled border resorbs via carbonic anhydrase.
That brings us to a perfect point for a rapid consolidation of what we've covered so far.
Right. Time for our first mid-episode recap. If you are navigating a tricky roundabout, or you just zoned out for 5 minutes, we are going to reset and summarize the foundation we've built, and we will hit the facts twice.
Takeaway 1 revolves around the ingredients. Bone is 70% mineral, and type I collagen, which is the only collagen that mineralizes, makes up 90% of the organic matrix.
That's the recipe.
Takeaway 2 covers the cells. The osteoblast builds and is the sole cell that mineralizes, originating from the mesenchyme. The osteoclast demolishes, originating from the monocyte macrophage bloodline.
And takeaway 3.
Takeaway 3 details the osteoclast machinery. The clear zone clamps using integrins, creating the seal. The ruffled border resorbs using carbonic anhydrase to pump acid.
Running through that one more time, rapid fire, 70% mineral. Type 1 collagen is 90% of the organic matrix and is the only one that mineralizes. Osteoblast builds and solely mineralizes from mesenchyme. Osteoclast demolishes from the bloodline. Clear zone clamps via integrins. Ruffled border resorbs via carbonic anhydrase.
With the foundation laid, we can now explore how these cells actually communicate.
We have our builders and our demolishers, but they need a molecular language to coordinate the tug-of-war. This brings us to the master switch.
A master switch, yes.
There is a trio of molecules here that unlocks a vast number of exam questions, but they are incredibly easy to confuse because the names are almost identical. Rank, rank L, and OPG.
The similarity in nomenclature is the trap. We're going to separate them distinctly, starting with rank.
Okay, let's start with rank.
Rank is a receptor protein. I want you to visualize it as a physical lock. This lock is located on the surface of the osteoclast precursor.
The precursor.
Yes. That precursor is the young, immature macrophage that has not yet fused and transformed into a bone-eating machine.
So rank is the lock, and it sits on the young, immature demolisher. What is rank LL?
Rank LL, spelled RANKL, stands for rank ligand. In biochemistry, a ligand is simply the key that fits into a specific receptor. Therefore, rank LL is the key that fits perfectly into the rank lock.
Okay, lock and key.
And here is the genius of the entire biological system. The rank L key is manufactured and held by the osteoblast.
Wait, let me deduce the logic there. If the builder holds a key and the lock is on the demolisher, then the demolisher cannot activate itself.
Exactly.
The builder literally has to hand the key over to turn the demolition crew on.
That is exactly the logic. The osteoblast is the control tower. The exam tests this concept relentlessly. When the osteoblast decides that bone needs to be resorbed, it presents the rank LL key.
And when it plugs in?
When that rank LL key plugs into the rank lock on the young osteoclast precursor, a massive intracellular signaling cascade is triggered. The precursors fuse together, become a giant multinucleated cell, develop a clear zone and a ruffled border, and become a highly active, mature osteoclast.
It is brilliant because it prevents runaway bone destruction. The immune-derived osteoclasts can't just go rogue. They need constant stimulation from the mesenchyme-derived osteoblasts.
Precisely.
But even with that control, there has to be a brake system. I mean, if the osteoblast just kept presenting the RANKL key, the osteoclast would eventually eat the entire skeleton.
There is a highly effective brake, and it is the third molecule in our trio, OPG, which stands for osteoprotegerin. The name translates to bone protector. OPG functions as a molecular decoy.
A decoy, like a flare drawing a heat-seeking missile away from an aircraft.
That is the perfect visualization. OPG is a soluble, fake receptor. It is secreted by the osteoblast and floats freely in the microscopic space between the cells.
So it's just floating around out there.
Right. Structurally, the OPG molecule looks almost identical to the rank lock. So, as the osteoblast releases RANKL keys into the environment, many of those keys get tricked. They bind to the free-floating OPG decoy instead of the real rank receptor on the osteoclast precursor.
So OPG intercepts the key. It soaks up the RANKL before it can ever reach the osteoclast, which means the osteoclast never gets the signal to mature.
Precisely. It is a game of molecular ratios. If the osteoblast secretes a massive amount of OPG, most of the RANKL keys are intercepted, very few osteoclasts mature, and bone resorption halts. The bone gets stronger.
And if it drops the OPG.
If the osteoblast stops producing OPG, the RANKL keys have a clear path to the osteoclasts, resorption ramps up, and bone is lost.
You know, I've seen a very specific, fiddly true-false question regarding OPG that traps candidates. The question lists two options regarding OPG's function. Option A says OPG inhibits osteoclast formation. Option B says OPG binds RANKL. From a clinical perspective, both of those statements are technically true.
They are both true in the broad sense, which is what makes it an excellent discriminator, but the exam demands precision regarding molecular biology. Binding RANKL is the mechanism of action. Inhibiting osteoclast formation is merely the downstream result of that mechanism.
So which do you pick?
In the FRCS, you must always select the precise mechanism. OPG binds rank well. And I must issue a strict warning here. Some older textbooks will sloppily state that OPG binds to rank. It absolutely does not.
It binds the ligands.
Yes, OPG is a decoy receptor. It binds the ligand. It binds the key, not the lock.
Mechanism over result. OPG binds rank well. Now, is RANKL the only signal required to create a mature osteoblast, or is it part of a combination code?
It is a combination code. The precursor requires a second signal called MCSF, which stands for macrophage colony stimulating factor.
MCSN.
The complete recipe for an active osteoclast is RANKL plus MCSF. MCSF provides the baseline survival and proliferation signals for the macrophage lineage, while RANKL provides the specific instruction to differentiate into a bone-resolving cell.
Let's log this trio in. The rhythm here is crucial. RANK is on the precursor. RANKL is from the osteoblast. OPG is the decoy that binds RANKL.
That's the rhythm.
Saying it again for the listeners. RANKL's on the precursor. RANKL is from the osteoblast. OPG is the decoy that binds RANKL out.
To solidify this, we really need to bridge this basic science into clinical pharmacology. The drug denosumab, commercially known as Prolia, revolutionized osteoporosis treatment. If you understand the RANK pathway, denosumab's mechanism of action becomes completely obvious.
Oh, denosumab. It's given as a subcutaneous injection every 6 months, right?
Yeah.
How does it interact with this master switch?
Denosumab is a fully human monoclonal antibody designed to target and neutralize ERNKL.
So it's an antibody against the key.
Exactly.
Oh.
It is, quite literally, an artificial version of OPG. When you inject denosumab into a patient, those antibodies flood the bone microenvironment and act as millions of synthetic decoys. They mop up all the available ERNKL keys, preventing them from activating the osteoclasts. Resorption is shut down, and the bone density increases.
That is incredibly elegant. The pharmacologist just copied nature's brake system. Denosumab is artificial OPG.
It's exactly that.
This brings us to the vital takeaway for the master switch. RANK is on the precursor, RNKL is from the osteoblast, OPG is the decoy that binds RNKL, and denosumab is simply artificial OPG.
Say it one more time.
Repeating that complete sequence. RANK is on the precursor, RNKL is from the osteoblast, OPG is the decoy that binds RNKL, and denosumab is artificial OPG.
Now that we understand the molecular switches located on the bone surface, we can finally zoom out and look at the systemic hormones that travel through the blood to flip those switches.
This is where systemic physiology meets bone biology. We will start with parathyroid hormone, or PTH, because it features what I consider the greatest logical trap in the entire basic science syllabus.
The PTH trap. It gets everyone.
It does. If you put yourself in the mind of a candidate reading a multiple-choice STEM under pressure, the logic seems obvious. The question asks, where is the primary receptor for parathyroid hormone located? The candidate knows that TTH's primary job is to mobilize calcium from the bone into the blood, which requires bone resorption.
Right.
Therefore, the brain deduces. If PTH drives bone eating, its receptor must be on the bone eater. It must be on the osteoclast.
It is a perfectly logical deduction, and it will cost you the mark every single time.
Every time.
The physiology is counterintuitive, but it enforces the rule we established earlier. The PTH receptor is located on the osteoblast. It is also found on the cells of the distal tubule in the kidney. But you must imprint this fact. The osteoclast has 0 PTH receptors, none whatsoever.
If the osteoclast is completely blind to PTH, how on earth does PTH cause massive bone resorption?
It operates exclusively through the control tower.
The osteoblast.
Yes. When PTH levels rise in the blood, the hormone binds to the receptors on the osteoblast. The osteoblast receives the systemic command to release calcium, and it executes that command by dramatically altering its molecular output.
What does it alter?
The PTH-stimulated osteoblast upregulates its production of the RNKL key and simultaneously downregulates its production of the OPG decoy.
Oh, I see the mechanics of that. It floods the area with keys and removes all the decoys. The osteoclasts are suddenly fully activated, not because PTH touched them, but because the osteoblast commanded them to activate.
Precisely. Furthermore, the PTH-stimulated osteoblast also secretes inflammatory cytokines like IL-6 and releases neutral proteases.
Neutral proteases.
Remember the protective tarp of unmineralized osteoid we discussed? Those neutral proteases help strip that tarp away, rolling out the red carpet for the activated osteoclast to dock and begin their destruction.
So if a question asks, PTH stimulates which cell to release neutral protease, the answer isn't some inflammatory immune cell.
It is the osteoblast. Almost every mechanism regarding PTH in the bone points back to the osteoblast.
Okay, that covers the cellular mechanism within the bone microenvironment, but we need to look at the net effect on the systemic blood levels. If a patient develops a parathyroid adenoma that is constantly pumping out PTH classic primary hyperparathyroidism, what happens to their serum calcium and phosphate levels?
The clinical effects are stark. PTH raises serum calcium and it lowers or wastes serum phosphate.
It wastes phosphate.
That second point is the critical exam discriminator. PTH is highly phosphaturic. It actively forces your kidneys to dump phosphate into the urine.
Wait, let me work through the logic there. If PTH is driving osteoclasts to dissolve hydroxyapatite, it is releasing both calcium and phosphate from the bone into the blood simultaneously. Why would the body want to keep the calcium but throw the phosphate in the trash?
It is a matter of physical chemistry. If PTH raised both calcium and phosphate in the blood simultaneously to high levels, those two ions would simply bind together in the bloodstream and precipitate out as solid calcium phosphate crystals in your soft tissues and blood vessels.
Oh, ectopic calcification.
Exactly. To prevent that, PTH orchestrates a complex routine. It pulls both from the bone, but then tells the kidney to save the calcium and dump the phosphate.
That explains the physiology perfectly. So the mnemonic we need to drill is PTH raises calcium, wastes phosphate.
That's the one.
Let's say it twice.
Yes.
PTH raises calcium, wastes phosphate. Now, how exactly does the kidney pull off that juggling act?
It utilizes different segments of the nephron. In the distal convoluted tubule of the kidney, PTH significantly boosts calcium reabsorption. It claws the calcium out of the urine and pulls it back into the blood.
And the phosphate.
Conversely, in the proximal tubule of the kidney, PTH blocks the reabsorption of phosphate, ensuring it is flushed out of the body.
So that's 3 moves. It pulls both from the bone via the osteoblast, saves calcium in the distal tubule, and dumps phosphate in the proximal tubule. Is there a 4th move?
The 4th move is arguably its most important systemic function. In the kidney, PTH acts as a switch to turn on an enzyme called 1-alpha hydroxylase.
1-alpha hydroxylase.
Yes, this is the specific enzyme responsible for activating vitamin D.
Does PTH have any direct ability to absorb calcium from the food sitting in the gut?
Absolutely not. And that is a classic false option on the exam. PTH's effect on the gastrointestinal tract is entirely indirect. It tells the kidney to manufacture active vitamin D, and it is the vitamin D that travels to the intestine to handle the dietary absorption.
Ah, so they operate as a biological relay team.
Exactly. PTH runs the first leg of the race, passes the baton by activating 1-alpha hydroxylase, and vitamin D runs the anchor leg in the gut.
Which leads us to the vital takeaway for this section. If you only remember one thing about parathyroid hormone, PTH's receptor is on the osteoblast and the distal kidney, never the osteoclast, and it raises calcium while wasting phosphate.
Definitely repeat that one.
Doubling down on that, PTH's receptor is on the osteoblast and the distal kidney, never the osteoclast, and it raises calcium while wasting phosphate.
That complex interaction between the master switch and PTH is a lot to hold in working memory.
It really is. Which means it is time for our second mid-episode recap. Let's consolidate the master switch and PTH before we move into vitamin D.
First, the rank trio. Rank is the receptor lock on the precursor. ARNKL is the key made by the osteoblast. OPG is the decoy that binds ARNKL, intercepting the key and stopping resorption.
Second, parathyroid hormone. PTH acts entirely through the osteoblast control tower. It never touches the osteoclast. It instructs the osteoblast to push ARNKL up and OPG down. In the systemic circulation, it raises calcium and wastes phosphate while switching on active vitamin D in the kidney.
Let's run that summary one more time for the drivers.
Rank on precursor. ARNKL from osteoblast. OPG the decoy. PTH acts in the osteoblast, raises calcium, wastes phosphate.
The unifying theme here is control. Whether it is a local signal or a systemic hormone like PTH, the osteoblast is the central command station receiving the data.
Which transitions us perfectly into the anchor leg of the relay race, vitamin D. This is an area where the nomenclature can cause genuine anxiety in an exam setting.
Oh, the numbers.
Yeah, there are 3 different chemical versions of vitamin D. They're all identified by numbers, and they look incredibly similar on a multiple-choice page. Let's systematically untangle the metabolites.
The easiest way to master vitamin D is to visualize it as a product moving down a biochemical assembly line, passing through 3 distinct organ stations in your body.
Okay, an assembly line. What's station 1?
Station 1 is the skin. Deep in the epidermal layers, you have a precursor molecule called 7-dehydrocholesterol. When sunlight, specifically ultraviolet B radiation, strikes your skin, the energy converts that precursor into vitamin D3, also known as cholecalciferol.
Okay, station 1 is the skin.
Yeah.
Sunlight makes D3. Where does it go next?
It enters the circulation and travels to station 2, the liver. The liver is the slow, steady processing plant. It takes that D3 and adds a single hydroxyl group specifically to the 25th carbon position. The output is 25-hydroxyvitamin D.
Station 2, the liver, creates 25-hydroxy.
Finally, it travels through the blood to station 3. The kidney. The kidney acts as the on-demand activation switch. It takes the 25-hydroxy, and using that enzyme we just mentioned, 1-alpha-hydroxylase, which is stimulated by PTH, it adds a second hydroxyl group at the first carbon position.
And that creates?
This creates 1025-dihydroxyvitamin D. That is a fully active, highly potent hormonal form.
So the assembly line is skin, liver, kidney. And the numbers climb sequentially, 25 after the liver, 125 after the kidney.
Exactly. The liver puts on the 25, the kidney adds the 1. Once you have that pathway visualized, we can navigate the 3 major exam traps hidden within it.
Let's hit the traps.
Trap number 1 is clinical. Which form of vitamin D do you actually request from the lab when you want to check a patient's nutritional status?
My instinct, if I'm on the ward and I want to know if a hormone system is functioning, is to measure the active version. I would guess we measure 1025.
It is a logical guess, and it is wrong. You never measure the active 1025 to determine a patient's baseline vitamin D stores. You always measure 25-hydroxyvitamin D, the intermediate form produced by the liver.
Wait, why measure the inactive intermediate?
Because of pharmacokinetics. The 25-hydroxy form generated by the liver serves as the body's storage pool. It has a long half-life in the blood, measured in recs, meaning its levels are stable and accurately reflect your true nutritional status over time.
And the active form?
The active 125 form produced by the kidney has a very short half-life, just a few hours. Furthermore, its production is tightly regulated by PTH. If a patient is severely vitamin D deficient, their PTH levels will spike to compensate, which will drive the kidney to convert every last drop of 25-hydroxy into the active 125 form.
Oh, so a deficient patient might show normal levels of the active form.
Yes, a severely deficient patient might actually have normal or high levels of 1025 for a brief period. It is a chaotic snapshot. To know the true reserves, you measure the stable pool. The rule is measure 25 for stores.
Measure 25 for stores. That makes perfect clinical sense. Now let's look at trap number 2. What does the active form, the 1025, actually do to the calcium and phosphate levels in the blood?
This is the punchline of the entire calcium homeostasis section. Active vitamin D raises both calcium and phosphate.
It raises both of them simultaneously.
It raises both. Active vitamin D is the great gut absorber. It travels to the small intestine, opens the doors, and aggressively pulls both calcium and phosphate from your dietary intake into the bloodstream. This is the crucial discriminator that separates it from PTH.
Right. You established earlier that PTH wastes phosphate to prevent ectopic calcification. If vitamin D raises both, why doesn't that cause the same dangerous crystallization in the blood?
Because the physiological goal of vitamin D isn't just to keep blood calcium stable, its goal is to provide the raw materials necessary to build and mineralize new bone. Hydroxyapatite requires both calcium and phosphate.
If you want to pour concrete, you need all the ingredients.
Exactly. Vitamin D supplies them. But for the exam, you must keep the hormone effects distinct.
Let's line them up so they are impossible to cross. PTH, calcium goes UP, phosphate goes down. Active vitamin D, calcium goes UP, phosphate goes UP. They agree on calcium, but they are total opposites on phosphate. In fact, active vitamin D is the only ITLI hormone in this entire syllabus that raises phosphate.
That fact is so heavily tested, it requires double repetition.
I'm on it. Active vitamin D is the only hormone that raises phosphate. Active vitamin D is the only hormone that raises phosphate. That is a massive key for the multiple choice questions.
There are 2 rapid physiological footnotes regarding the gut absorption that the examiners occasionally deploy. First, where exactly in the gut does this occur? It is the proximal small intestine, specifically the duodenum and jejunum.
Proximal small intestine, got it.
Second, the calcium doesn't just float across the intestinal cell membrane passively. It requires a physical delivery truck to haul it across the cell against a concentration gradient. That delivery protein is called calbindin. And the production of calbindin is directly stimulated by vitamin D. So vitamin D doesn't just unlock the door, it actively manufactures the delivery trucks.
That is a brilliant level of detail. Now, you mentioned a third trap. We've covered 25 and 1025. What is the third trap?
The trap revolves around a third metabolite, 24,025-dihydroxyvitamin D. Historically, older physiology textbooks dismissed this molecule as simply an inactive waste product, a way for the kidney to safely dispose of excess vitamin D.
But I'm guessing it's not just waste.
Nope. Modern research has revealed a specific function, and the exam has begun testing it. 24,025-dihydroxyvitamin D acts as a mitogen in the proliferative zone of the growth plate. It actively stimulates the chondrocytes, helping growing children's bones mature, and it is implicated in fracture healing.
That is incredibly sneaky. If an exam stem presents a pediatric patient and specifically asks which vitamin D metabolite drives growth plate chondrocyte maturation, the instinct is to pick the active 1005. But the correct answer is the seemingly inactive 24,025.
Exactly. It is a perfect discriminator between candidates who are up to date and those relying on outdated summaries.
I am going to set the three forms to a rhythmic mnemonic and repeat it twice so it survives the drive. 25 for stores, 1,025 for action and raising phosphate, 24,025 for the growth plate.
Again?
One more time. 25 for stores, 1,025 for action and raising phosphate, 24,025 for the growth plate.
Internalizing that rhythm will confidently secure you several marks.
Which brings us to the vital takeaway for this assembly line. If you only keep one thing from this segment, 25-hydroxy tells you the stores. Active 1,025 is the only hormone that raises phosphate. Again, 25-hydroxy tells you the stores. Active 125 is the only hormone that raises phosphate.
We have spent a lot of time discussing how the body raises calcium levels. PTH pulls it from the skeleton. Vitamin D absorbs it from the gut. But this is a homeostasis module. What goes up must come down. If blood calcium levels spike dangerously high, who pushes calcium back down?
We need an emergency brake. What pulls that brake?
The emergency brake is calcitonin, and calcitonin is conceptually the mirror image of almost everything we have discussed so far. First, let's establish its origin. It is synthesized and secreted by the parafollicular C cells of the thyroid gland.
C cells.
A very handy mnemonic is simply C cells for calcitonin. It is released directly into the bloodstream in response to acute hypercalcemia when blood calcium gets dangerously high and threatens to cause cardiac arrhythmias or neurological depression.
C cells for calcitonin, and its job is to lower calcium rapidly. But how does it execute that? Does it travel to the osteoblast control tower and ask it to stop producing NKL the way PTH asks it to start?
This is where calcitonin becomes a beautiful, testable exception. It proves the fundamental rule by breaking it. We established earlier that the osteoclast, the demolition cell, has no PTH receptors. It ignores systemic hormones and only takes localized orders from the osteoblast.
Right.
Calcitonin is the single sole exception to that rule. Calcitonin has a receptor directly on the osteoclast.
Wait, it skips the control tower entirely. It doesn't even talk to the osteoblast.
It bypasses the control tower completely. Think about the physiological necessity. If calcium is dangerously high, threatening the heart, the body does not have the luxury of time. It cannot wait for the osteoblast to slowly alter its gene expression, decrease NKL, increase OPG, and let the osteoclast slowly die off.
It needs to stop now.
Right. Calcitonin plugs straight into the calcitonin receptors on the active osteoclasts and immediately paralyzes them. The ruffled borders retract, acid pumping ceases, and bone resorption halts instantly.
Yeah.
Over a longer period, it also decreases the formation of new osteoclasts.
So out of all the molecules in this vast syllabus, calcitonin is the one and only systemic hormone that acts directly on the osteoclast.
It is the ultimate discriminator. Which of the following acts directly on the osteoclast? The answer is always calcitonin. Additionally, it acts on the kidney to increase the excretion of calcium into the urine. The combined rapid shutdown of bone resorption and renal dumping causes serum calcium to fall rapidly.
Does this have an application in modern clinical orthopedics?
We utilize it in specific high-turnover scenarios. If a patient presents with acute, severe hypercalcemia of malignancy, or if they have incredibly painful acute osteoporotic vertebral compression fractures, we can administer calcitonin. It rapidly shuts down the rampant bone turnover, which not only lowers the calcium, but can also provide significant analgesic relief for bone pain.
The mnemonic for this hormone's function is incredibly straightforward, and we will drill it twice. Calcitonin tones calcium down. Calcitonin tones calcium down.
It tones it down, secreted from the thyroid C cells, bypassing the builder and acting straight on the osteoclast.
The vital takeaway for the emergency brake. Calcitonin lowers calcium, comes from thyroid C cells, and is the only hormone that acts directly on the osteoclast. One more time. Calcitonin lowers calcium, comes from thyroid C cells, and is the only hormone that acts directly on the osteoclast.
Now, to tie a definitive bow on this entire systemic tug-of-war, we need to ask one final critical question. What happens to the skeleton when the most protective, overarching hormone of all suddenly disappears?
This is where basic science translates directly into the most common metabolic bone condition we treat: postmenopausal osteoporosis. The missing protective hormone is estrogen. What is estrogen actually doing to the bone microenvironment on a daily basis in a healthy adult?
Estrogen is profoundly pro-bone, and it exerts its protection in every possible direction simultaneously. I want to bring back the rank master switch we discussed earlier because estrogen pulls that specific lever very hard.
Okay, how so?
Estrogen acts on the osteoblast control tower, commanding it to massively decrease the production of the AR-NKL key while simultaneously upregulating the production of the OPG decoy.
So it suppresses the key and floods the area with decoys.
Exactly. The net result is that very few osteoclasts ever mature. But estrogen's protection doesn't stop there. It also acts directly on mature osteoclasts to actively push them toward apoptosis, which is programmed cellular death. It limits their lifespan.
Wow. It attacks them on all fronts.
It does, and simultaneously it protects the osteoblasts and the buried osteocytes from apoptosis, ensuring they live longer, productive lives. Finally, it suppresses the background production of inflammatory cytokines like IL-1, IL-6, and TNF, all of which normally stimulate bone resorption. Estrogen is an absolute, multifaceted shield for the skeleton.
If that is the baseline state, walk me through the pathophysiology of menopause. When that shield drops, what happens at the cellular level?
At menopause, circulating estrogen levels plummet, effectively falling off a cliff. The sudden withdrawal of that shield throws the microenvironment into chaos. Without estrogen holding them back, the osteoblasts suddenly begin pumping out massive amounts of AR-NKL, and they drastically drop their OPG production.
And the cytokines.
The inflammatory cytokines surge. The mature osteoclasts live much longer because the signal forcing them into apoptosis is gone. The entire metabolic tug-of-war tips violently and aggressively toward breakdown.
The demolition team is handed unlimited keys and no brakes.
That is the exact cellular mechanism of postmenopausal rapid bone loss. It perfectly explains why hormone replacement therapy was historically such a potent protective agent against osteoporotic fractures, and why losing it is so devastating to trabecular bone architecture.
Which brings us to the grand convergence of this entire module. If we step back and look at the big picture, we can logically group all the major anti-resorptive agents we've covered today. They all achieve the exact same macroscopic goal, but they use completely different biochemical doors to get there.
They do, and realizing this is the most satisfying aha moment of the revision process. If you line up the anti-resorptive team, native OPG achieves it by acting as a decoy and soaking up the AR-NKL key. Estrogen achieves it by commanding the osteoblast to drop the key and raise the decoy.
And calcitonin.
Calcitonin achieves it by pulling the emergency brake on the osteoclast directly. And denosumab achieves it by being an artificial OPG decoy injected from a syringe.
That leaves the most famous osteoporosis drug class of all, one that every orthopedic trainee prescribes constantly. The bisphosphonates, elendronate, zoledronate. Where do they fit into this molecular convergence? Because they don't target AR-NKL, do they?
Bisphosphonates achieve the exact same goal, paralyzing the osteoclast, but they attack from a completely different, ruthless angle. This mechanism is highly examinable because it is unique. Bisphosphonates do not circulate and bind to a receptor on a cell surface.
How do they go?
They bind directly and with extremely high affinity to the hydroxyapatite crystals in the bone matrix itself.
Wait, they bind directly to the concrete. They become part of the structure.
They embed themselves physically into the mineral. They just sit there silently waiting. Then, when an active osteoclast comes along, clamps down its clear zone and begins pumping acid from its ruffled border to dissolve that specific patch of mineral, it unwittingly dissolves and swallows the bisphosphonate drug along with the calcium.
It eats a poisoned apple.
Exactly. Once the bisphosphonate is internalized inside the cytoplasm of the osteoclast, it acts as a devastating cellular poison. Specifically, and this is for the high-achieving candidates, the nitrogen-containing bisphosphonates disrupt a critical internal metabolic chain called the mevalonate pathway.
The mevalonate pathway.
They block an enzyme called farnesyl pyrophosphate synthase. Without that pathway, the osteoclast cannot maintain its ruffled border, it cannot traffic vesicles, and it rapidly undergoes apoptosis and dies.
That is pharmacological sabotage. The drug poisons the very crystal the cell is designed to eat.
It is a brilliant, highly targeted trap. Because the drug only goes where the bone is actively being chewed up, it has minimal effects on resting bone. But for this FRCS module, the core examinable mechanism you must commit to memory is the binding site. Bisphosphonates do not bind to a gut receptor, they don't bind to a kidney enzyme, and they don't bind to rank. They bind hydroxyapatite and kill the osteoclast.
We will repeat that mechanism twice. Bisphosphonates bind hydroxyapatite and kill the osteoclast. Bisphosphonates bind hydroxyapatite and kill the osteoclast. And the vital takeaway to close out this final systemic section, estrogen protects bone by dropping ORNKL and raising OPG. Its disappearance in menopause lets resorption win. Again, estrogen protects bone by dropping ORNKL and raising OPG. Its disappearance at menopause lets resorption win.
That completes the landscape. We have traced the thread from the ingredients of the concrete, through the cells that build and destroy it, up to the hormones and drugs that dictate the battle.
Which means we have arrived at the final lightning round. If you are a candidate doing the dishes or circling the hospital car park right now, this is the rapid-fire consolidation that cements it in your brain. We are going to do one fast lap of the highest yield discriminators. I will say the prompt, you deliver the exam answer. Ready?
I'm ready. Let's do it.
What is the composition of bone?
70% mineral, 22 to 25% organic, 5 to 8% water. Type 1 collagen is 90% of that organic matrix and is the only collagen that mineralizes.
Which specific cell mineralizes bone?
The osteoblast, solely.
On the osteoclast. Clamp versus destroy.
The clear zone clamps via integrins. The ruffled border resorbs via carbonic anhydrase. Attachment is always the clear zone.
Define the rank trio.
Rank is the lock on the precursor. Rank L is the key from the osteoblast. OPG is the decoy that binds rank L.
Where is the PTH receptor?
On the osteoblast and the distal kidney, never on the osteoclast.
Compare PTH versus active vitamin D regarding phosphate.
PTH wastes phosphate. Active vitamin D raises phosphate.
What are the traits of calcitonin?
Secreted from thyroid C cells, lowers calcium rapidly, acts straight onto the osteoclast.
What is the mechanism of a bisphosphonate?
Binds directly to hydroxyapatite, poisons, and kills the osteoclast.
We have successfully untangled the web. The avalanche of facts is now a single logical story.
As a final provocative thought, I want to leave you with the ultimate heuristic for tackling this specific module on exam day, building on everything we have just mapped out. Whenever you read an exam question that tries to make the osteoclast the primary listener, the cell that is supposedly hearing a grand systemic hormonal message, your baseline reaction should be deep suspicion.
Be suspicious of the osteoclast.
Always. Step back and question the premise. Because the fundamental, inescapable rule of bone biology is that the message almost always goes to the control tower. It goes to the osteoblast. The builder controls the demolisher. Calcitonin is the one isolated, beautiful exception that proves that magnificent rule. Keep that heuristic in your pocket, and many of the tracks will simply disarm themselves.
The murky waters of that diagnostic X-ray finally cleared up by understanding the microscopic tug-of-war beneath the surface. That brings us to the end of Module 1. Next time on our deep dive, we are taking this exact cellular foundation and applying it to the pathology of metabolic bone disease. We will cover the crucial biochemical differences between osteoporosis and osteomalacia, dissect the wild bone turnover of Paget's disease, dive deeper into the varied causes of hypercalcemia, and explain exactly why chronic steroids wreck bone architecture. The clinical stakes get higher, but the players are exactly the same. It is the same tug-of-war. Until then, keep that structure in mind, and best of luck with the revision.
Keep up the hard work. We will be here when you are ready for the next module.